Hello everyone. Today's lecture is about cell chemistry, and we talk about main building blocks: protein, lipids and polysaccharides. So before we start to go into the cell chemistry, I want just to remind some of you that the information about all chemical components in cells like proteins and polysaccharide and lipids is programmed in the DNA. Well, actually, it's not completely true, because all the main information in which DNA is carrying is related to proteins, and then proteins are doing all of the work related to the synthesis of the lipids and polysaccharides. So, if you look in the cell. In any cell you have a DNA, which in eukaryotic cells is packed in nucleus and chromosomes. In prokaryotic cells it has much simpler organization, just as in an unpacked form. So the DNA is carrying information about proteins. Different types of proteins can be synthesized in the cell, and they have, of course, different structure, which we will look more in detail now. So proteins, that's the high molecular organic substances, so-called biomolecules or micromolecules, consisting of one or more long chains of amino acid residues. It means that the building blocks of protein is amino acids. Amino acids are organic compounds which contains amine and carboxyl functional groups. So usually we have on one end of the amino acid we have amine group, and on the another end we have carboxyl group. In addition to these two groups, in all amino acids we have radicals, or so-called side chain groups. Here, as you can see, so these groups are related to different types of the radicals, and actually they are different in different amino acids. Yeah, so if we talk about amino acids, what we can say about their chemistry. So amino acids are crystalline substances, and most of them are soluble in water. And when they are dissolved in water, the carboxyl and amino groups of amino acids undergo partial ionization. It means that in the water solution of amino acids, we have molecules which are charged, and these charged molecules are capable of exhibiting both acid and alkali functions due to these two groups, amino and carboxyl groups. Currently, there is about 500 amino acids known, and most of these amino acids are actually chemically synthesized. But the most important amino acids which are found in cells, that's 20 amino acids. And these 20 amino acids actually encoded in the DNA. So all amino acids could be grouped into 4 groups, which is non-polar, polar, charged and non-charged, positively charged and negatively charged amino acids. Depending on the chemical structure of the radical group, of the side chain, we can identify these 4 groups. Amino acids are building blocks of proteins. And now I will show you how amino acids are connected to each other in the proteins. So, as I said, in the amino acids there is amine and carboxyl group, and these 2 groups, they form peptide bond. So when a peptide bond is formed, water is released. And the peptide bond connecting amino acid to each other, and by that we get a molecule which called peptide. The peptide has on one end it has amino group and it's called amino end, and on the another end it has carboxyl group and it's called carboxyl end. So peptides can have very long chain structure and they are unbranched. And such molecules are called polypeptides. Polypeptides, actually, they are the first level of protein organization. So all proteins has 4 levels of organization. First is primary protein structure, second is secondary protein structure, third is tertiary protein structure, and fourth is a quaternary protein structure. So, as I mentioned already, that the first primary protein structure is a polypeptide, so that's a long chains of connected amino acids, and these chains are unbranched, and the main bonds in the polypeptides is peptide bonds. Now we move to the secondary protein structure. The secondary structure of proteins is formed by the interaction between the amino acids of one polypeptide and amino acids of another polypeptide. And the bonds which are responsible for forming secondary protein structure is hydrogen bonds. So there are 2 types of secondary protein structure. It's alpha helix and beta pleated sheets. As you can see, morphologically they are quite different, but in both of them the main bonds which are formed is hydrogen bonds. The third structure of proteins is tertiary protein structure. The tertiary protein structure of the protein is even more complex. In the formation of this structure, there are several types of bonds and chemical interactions are involved. The main types are covalent bonds, and that's so-called, I can show now, this is so-called disulfide bridges. This is covalent bonds. So these disulfide bridges, they are formed between different amino acids. Also ionic bonds between oppositely charged side groups of amino acids, hydrogen bonds, as it was in case of the secondary structure, and hydrophobic interactions. That's what you can see. The tertiary structure determines the spatial organization of the protein to the greatest extent. So for many proteins, the tertiary structure, that's actually the ultimate level of the protein organization. And in this form, most of the proteins, they are showing their highest activity and they are performing their functions. Some proteins are having further level of organization as quaternary structure. So When we study proteins, since we see that proteins have quite complex organization structure, so very often we use 3D imaging to study proteins to understand all these structures. And to study 3D organization of proteins, we using so-called three-dimensional models. And there are several options to study three-dimensional structure of the protein. Here I show 3 examples of these. On the left side you see so-called all-atom representation colored by atom type of the protein. In the middle side you see simplified representation illustrating the backbone conformation colored by the secondary structure. And in the right side you see solvent accessible surface representation colored by residue type. So you can see that acid residues are colored by red, basic residues colored by blue, polar residues colored by green, and non-polar residues are grayish. Quaternary structure, as I said, not all proteins have the quaternary structure, but some of them have this level of organization. So quaternary structure is obtained by combining several polypeptide chains in a single protein complex. The structure of the protein with quaternary structure can include both identical and different polypeptide chains. An example of such a protein could be hemoglobin. In the stabilization of this level of organization, the same type of connections and chemical bonds are involved as in the tertiary structure. So it's sulfuric bridges, hydrogen bonds, and covalent bonds, and also hydrophobic interactions. When we are discussing the structure organization of the proteins, there are 2 processes which is important to mention. That's denaturation and renaturation of the proteins. So denaturation of the proteins, that refers to any changes in the protein molecule associated with the loss of the quaternary or tertiary or secondary structure. That's what you can see here. So the proteins denaturated. The renaturation is related to the process when the proteins are going back to its secondary, tertiary or quaternary structure. So such changes usually occur under the influence of various destabilizing factors, depending on the nature of denaturating agent. For example, mechanical agents like strong mixing or shaking, physical agents as heating, irradiation, and sonication, and chemical agents like acid and alkali. Denaturation is happening. Examples of the denaturation could be frying eggs. So when we are frying eggs, we denaturate in our proteins, and that's irreversible process, so we can't renaturate the proteins back. And as example of the denaturation, that's boiling milk. So when we are cooking milk, all the proteins in the milk are also denaturating. So it was shortly about the chemistry of the proteins and the structure, and now I want to talk more about the diversity of the protein functions. So actually protein in the cells are carrying many different functions. So the first function, and which is one of the most important functions of the proteins in all living organisms, is catalytic function. So absolutely all living organisms and all biochemical processes which are happening in the living organisms associated with the catalytic function of the proteins. And the proteins which are doing catalytic function are called enzymes. So enzymes catalyze reactions of cleavage of complex molecules and the synthesis of the complex molecules. And they're involved in the metabolism, in the anabolism and catabolism. Many of these reactions are well studied, and they're actually very complex reactions. But there are many reactions which are catalyzed by enzymes which are still not discovered. As you can see here, that's kind of simplified map of several main biochemical reactions which could be catalyzed by the proteins. The next function of the proteins, that's a structural function. Proteins are part of the cytoskeleton of the cell. They are also responsible for shaping many organelles and involved in changing of the shape of the cell. So as you can see, they're also part of our hairs, they're part of the horns and feathers of animals. And the example of two such proteins is collagen and elastin. That's the two main proteins of intracellular substance of connective tissue. Examples of another proteins is keratin, and which is part of our hairs, nails, feathers, and shells, for example, for the eggshells. The next function, which is also important to mention, that's the protective function. So proteins are a part of blood and other biological fluids, and they participate in protecting our body from pathogenic bacteria and viruses. An example of such proteins is antibodies or immunoglobulins, which are neutralizing bacteria, viruses, or foreign proteins, providing the body immunity. So all our immune system is based on the protective function of proteins. So proteins are protective proteins or immunoglobulins. They are able to bind the toxins from bacteria or other sources, and they can ensure the detoxification of these proteins. The next function is a transport function. So as I said, now that the proteins are part of the blood, and one of the most important protein in the blood, that's hemoglobin, which is responsible for transporting oxygen from lungs to tissues and CO2 from tissues to lungs. So and that's actually one of the important functions for keeping all cells and organisms in a live form. The next function is motor function. And there is a quite big class of motor proteins which provides the movements of our body and also body of animals and also some cells. So all our muscles are built up by the proteins, and there are two main muscle proteins in humans: it's actin and myosin. So also due to motor proteins, the cells in our bodies are able to move. For example, the movement of leukocytes. In some animals, like pseudopods, we also see motion proteins which are responsible for moving such organisms in the environment. The last function which I want to mention of proteins, that's a regulatory function. So the regulatory function of proteins, that's provides the ability for regulating some processes in our body. And here I put two examples. That's a human growth hormone and insulin. So both proteins are regulating different processes in the body. So as you can see, human growth hormone is regulating our growth. That's why we grow. And insulin actually involved in the digestion, and it's related to the consumption of the glucose by our body. So we are finished with the description about the proteins and their functions, and we are moving to the second group of organisms, to the second group of molecules, that's lipids. So lipids is a large and very diverse group of naturally occurring organic compounds that are related to their solubility in non-polar organic solvents, for example, chloroform, acetone, and benzene, and general insolubility in water. So, lipids could be grouped in three groups: the so-called simple lipids, compound lipids or lipids-like compounds, and derived lipids, or molecules which are derived from the lipids. Simple lipids, it's esters of fatty acids, and here we are talking about fats, that's esters of fatty acids and glycerol, and waxes, esters of long-chain fatty acids and long-chain alcohols. Compound lipids, that's the molecules which are also esters of fatty acids and alcohol chains, but they also contain other groups. Derived lipids, that's a compounds which is composed of hydrocarbon rings and a long hydrocarbon side chain. So all these three groups of lipids we can find in all living organisms. When we talk about fats, just one thing that I would like to mention, that fats, that's the most abundant group of all lipids you can meet in the cells, and esters of fatty acids and glycerols, they are common in the form of one huge molecule which called triglycerides. So in this molecule we have glycerol bond, and then we also have three fatty acid chains which are connected through the COC bonds. So fats, the examples of the fats where you can see, that's animal fats. So all the animal fats we eat, also all fat our body is able to generate and to store, that's in the form of triacylglycerides, where you have a glycerol connected to three fatty acids. Another example of this type of fat we see in the vegetable oils. So vegetable fats are a bit different from animal fats because they contain more unsaturated fatty acids than animal fat. The function of triacylglycerides is the following. So that's first of all, it's energy source. For sure, that's the body is using this fat for the energy when it's coming into the condition where there is no nutrients enough to consume. It's also heat insulator. It's also related to being a source of water and high flotation source. When we talk about waxes, so waxes, as I said, it's the esters of the long-chain fatty acid and long-chain alcohols. And waxes, the first example which comes into my mind, that's actually the waxes which are produced by bees. Also, waxes are very often we can find in plants. The function of the waxes is, first of all, structural, so it gives a structure or like a skeleton. It's also protective, so it protects from the unfavorable conditions of the environment. In addition to the protection, it's also reserve for the different nutrients. Compound lipids or lipids-like compounds, they have very complex structure. As you can see here in these molecules, we see polar groups and we see glycerol groups and free fatty acid. So usually in lipids-like compound we have two fatty acid bonds and we have polar group connected to one of the carbon of the glycerol. The polar group very often can contain phosphate group and also a radical group. Looking at this structure of the lipid-like compound, we can identify that phospholipids in the cells, they are related to this group of compounds. Phospholipids, they are part of the all cell membrane. So all cells of all living organisms, they are having phospholipids. And that's the very important structural component of the cell membrane. So the first function of these compounds is structural, of course. Also, there are compounds called glycolipids. As you can see, they have a bit different structure. We have sugars connected with the glycerol molecule, which is also connected to the fatty acids. So the function of the glycolipids is also structural because it's also part of many cell membranes. And the last group, derived lipids. So derived lipids, they are having, they don't have a free fatty acids part involved. As you can see, they have a lot of cyclic parts and different radicals, OH radicals, CH3 radicals, and O parts. So this, that's so-called derived lipids compounds. They are having a function of the a hormonal function. Also in plants, in higher microorganisms, and in animals. One of the examples, that's the steroids, that's the hormones in human bodies, but also in animal bodies, which is having different functions, and they are regulating different processes in our body. Another example is terpenes and carotenoids. That's also hormones, but they are also so-called pigments. So, for example, carotenoids, that's the orange pigments, which is actually responsible for the color of carrot. So carotenoids, they are having antioxidant function, so they are protecting our, they are able to protect the body against of the free radicals. The last group of molecules which we can find in the cells, that's polysaccharides. Polysaccharides are polymeric carbohydrate-based molecules which composed of the long chains of monosaccharide units bounded together. And after hydrolysis, polysaccharides, they usually give two types of molecules, monosaccharides and oligosaccharides. So here you can see three examples. Glucose, that's a monosaccharide. Sucrose, that's a disaccharide. And amylose. This is a polysaccharide, and actually that's the main component of starch molecule. Function of the polysaccharides, structural. Of course, polysaccharides are they are part of the plant cells, and they are part of the cell walls. So they are, together with the phospholipids, they are providing a structure of the cells. Example, one actually important example of such structural polysaccharide is histine. So histine is we can find histine in insects. So actually histine is making a structure of the insect's body. Also mushrooms. So mushrooms, they are rich in histine. And histine is responsible for forming structure of the mushrooms. Another function is reserve. Of course, when we talk about such polysaccharide as starch and glycogen, so that's the two reserve molecules which we find in living organisms. So starch you find in plants. Of course, examples is potato. That's where we find high content of starch. And glycogen we find in mushrooms and also in liver, for example. So in human body, glycogen is formed in liver. And that's together with the fat, it's also considered as a reserve for the energy. That was shortly about different three different groups of molecules in the cells, and I just want to mention how we study these molecules in the cells, because as you can imagine, all these molecules are very tightly connected to each other, and they are communicating with each other through the millions of chemical reactions. So there is a field called proteomics. That's a field of science where we study proteins. It's 3D structure organization, so all levels of structural organization. Also how the proteins are synthesized. The protein synthesis is called expression, so we are looking at when and how they are expressed in the cells, and we also are interested in the interaction of different proteins with each other in the cells. So proteomics is very tightly related to bioinformatics, because as a scientific field itself, proteomics is generating a huge amount of the data, which is very complex data, and they are and in order to analyze them, we need to use quite sophisticated data analysis tools which are developed by bioinformatics. To study lipid molecules and also polysaccharide molecules, that is happening in a scientific field called lipidomics. In lipidomics, we also connected very tightly to bioinformatics because of the amount of the data and the complexity of the data which we produce. And as you can see here, the example how we analyze lipids is that we start with a sample, and the sample need to be treated to extract the lipid fraction from the sample, and this lipid fraction further analyzed by the mass spectroscopy analysis, and if it's polysaccharide, it's analyzed by the HPLC analysis, and then the obtained data, they go through the very complex data analysis to extract the chemical information we are looking for. So this was a short overview about the cell chemical components, proteins, lipids, and polysaccharides. Also shortly about how we actually analyze these molecules. Thank you very much for your attention, and good luck.